Hello everyone, welcome to Medical Neuroscience and welcome to our first tutorial on the functional microanatomy of neurons. As we think together, I have three learning objectives for us to consider. First, I want you to be able to describe in very basic terms the classes of cells found in the central nervous system. I want you to be able to focus on one particular class that will spend much of our time in Unit 1 of this course considering, the neurons. And I want you to think about the functional microanatomy of neurons. Now, I'll spend this tutorial explaining what I mean by that, but essentially I want you to be able to differentiate between the different parts of a typical neuron in terms of their structure and their function. And then lastly, I want you to be able to describe how that microanatomical structure of a typical neuron is compounded many thousands of times into the structure of neural tissue. And when we think of neural tissue, we're basically talking about gray matter and white matter. Okay, well, let's begin by looking deep into the brain. And to do that, I'm going to show you a slide that I prepared some years ago through the motor cortex of the human brain. So we're looking at just a few hundred square microns of tissue that has been stained with a particular dye called thionine to reveal the presence of what we call Nissl substance, which is basically the rough endoplasmic reticulum, or the machinery that's making proteins within cells. So it's just a great way to look at the cells that are present in tissue and to appreciate something about the composition of this tissue. So we're looking here, and we can see a variety of different types of cells. And indeed, the brain is a very complicated place, even from a histological perspective. So there are a variety of types of cells here, and I think we can roughly categorize them into three types. There are neurons, which are the primary processors of neural signals. And as I mentioned, we're going to spend a lot of time together thinking about neurons. There are neuroglial cells, which would be the subject of the next tutorial. And neuroglial cells, or just glia for short, perform a rich variety of functions. And just to summarize those functions, I would say that they support the electrical and the chemical functions of neurons. And then, of course, the brain being a highly vascular structure requiring a constant supply of blood, and in that blood comes nutrients such as glucose, oxygen, and the ability to eliminate waste substances like carbon dioxide. So with that rich supply of blood that we find throughout the central nervous system, we'll find vascular endothelial cells in brain tissue as well. So let's focus now on a neuron, and I'll make a few general remarks first, and then we'll get on with our survey of the microanatomy of a typical neuron. So neurons are the fundamental unit of function in the central nervous system. This is a very important point, and this is the reason why we'll spend so much of our time and energy in Unit 1 thinking about how neurons actually function. Now, for those of you who have a bit more background in the field of neuroscience or are more in tune with contemporary thinking in the field of neuroscience, you'll realize that some might challenge this dogma that's been with us since the debate of Ramon y Cajal and Camilo Golgi more than a century ago. So with the more modern discussions for the aficionados acknowledged, I would continue to assert that the fundamental unit of function in the central nervous system is indeed the neuron. Now, neurons possess all the metabolic machinery that are common to other somatic cells. So there's a region of the neuron that contains the nucleus and many of the organelles that are necessary for cellular life. So in that respect, neurons are very much like other somatic cells. But in several important respects, neurons are quite distinct. Neurons come in a rich diversity of form, and we call that form morphology. So when one sees diversity of morphology, you should be thinking that there might also be diversity of function or physiology, and indeed that's the case, as form and function and structure are intimately connected within the central nervous system, even at the level of individual brain cells. Neurons have unique bioelectrical properties that distinguish them from most, but not all, other somatic cells. For example, the machinery that's necessary to generate electrical signals in muscle cells is quite similar to what we see in most nerve cells. And finally, there are specializations for intercellular communication that are pretty unique to the nervous system. And for many neurons, most neurons in the mature central nervous system, this involves the secretion of special chemical molecules called neurotransmitters. Now, again, neurons have not evolved this capacity independently of other kinds of cells in the body. There are other secretory cells that are well known in other kinds of tissues, but the challenge of intercellular communication demands some pretty interesting and some pretty unique specializations that we'll come to shortly. All right, now let's survey the different parts that make up a typical neuron, such as the one illustrated here, and this will give us a chance to think about what I consider to be the functional microanatomy of neurons. And let's begin with the cell body. So neurons have a cell body, which, as I mentioned, contains all the essential organelles, importantly including the nucleus, that are common to other somatic cells. And for most neurons, this cell body is actually quite rich in organelles because of the incredible synthetic capacities that are required for neurons to maintain their morphology as well as to support their function. So we find an abundance of endoplasmic reticulum, both smooth and rough, for example, for the synthesis of lipid molecules and proteins, respectively. We find an abundance of mitochondria for generating the enormous energy supply that's necessary to keep neurons functioning within their homeostatic states. So, for a variety of reasons, the cell body is a very important component of the neuron. Now, from that cell body grow out numerous different sorts of protoplasmic extensions. And I'd like to draw your attention to one set of extensions, which are typically short, about 100 microns in length, and they emanate in all different directions from the cell body. These are called dendrites. Now, dendrites are very important for the microanatomy of a neuron because what they do is that they extend the surface area that allows the cell to receive inputs from other neurons. So wherever we see dendrites, we think of this as an input zone because dendrites are the means by which cells receive synaptic contacts from other neurons. Now, some synaptic contacts are made directly onto cell bodies, but for the most part we find them on dendrites. Now, dendrites also have some very peculiar, very fine microstructural features, and these are called spines. So if I can illustrate just a length of a dendrite for you, I'll show you what I mean. Imagine that's a dendrite, and if we were to look at very high power, we might see under a microscope that there are these tiny mushroom-shaped protrusions out the side of that dendrite. These are called spines. Sometimes they're not quite so mushroom-like in their shape. They're more of a filament that extends some short distance away. And sometimes there seems to be a little stalk and a protrusion at the tip of that stalk. So collectively, these yet increase the surface area of even a single dendrite and provide a place for synaptic contact from other neurons. So wherever we see a spine, we can be quite confident that there is a synapse in one state of formation or another from the axons that are surrounding that dendrite. Now, not all dendrites grow out spines. Some dendrites are smooth. And where we see cells that grow out smooth dendrites, we know that these cells tend to be those that have an inhibitory effect on the cells that they make synaptic connections to. So the presence or absence of spines on dendrites is one means by which we can classify neurons. And we know that that classification based on the morphology of the dendrite gives us some insight into the functional properties of those cells. The morphology of dendrites is one way that we can differentiate various classes of neurons, even among those that do in fact grow out spines. One of the very interesting types of cells that we find in the cerebral cortex is called the pyramidal neuron. And it's called that because the cell body is shaped something like a little pyramid. And from that cell body grow out a series of dendrites. And from the apex of the pyramid is one rather thick, stout dendrite that we call the apical dendrite, because it grows out from the apex towards the surface of the brain. At the base of the pyramid are a variety of other dendrites called basal dendrites. And this special apical dendrite might have some unique properties with respect to its role in receiving signals from other neurons, and those might be somewhat distinguished from those in the basal dendritic tree. So the distribution of dendrites is again another important way that we have of classifying different kinds of neurons. Some neurons are multipolar, and these neurons give rise to dendrites that seem to just emanate in all different directions without necessarily growing out a single apical dendrite. So again, another example of how dendritic morphology can give us some insight into the structure and physiology of different cells in the brain. All right, so let's continue our survey. Now, growing out from the cell body is a very particular protoplasmic extension that has unique properties that allow it to generate electrical signals. And this process is called the axon. Now, what's illustrated here is a fairly simple picture of what a typical axon might look like. It's a single process often that grows out from the cell body and can grow for some distance. Some axons are short, being less than 100 microns, and others are very long. In fact, some are incredibly long. Consider, for example, a cell that might have its cell body in the motor cortex right about here inside your head, and that cell has to grow an axon all the way down through the brain, through the brainstem, and into the spinal cord. Perhaps some length in the spinal cord, maybe even to the lumbar or sacral segments of the spinal cord. So that could be maybe a half a meter or more, depending upon your height. Or consider the motor neuron in the spinal cord itself that's going to send an axon out in a spinal nerve to innervate a muscle. If you're a very tall person, that axon might be close to a meter in length. Really an amazing dimension, considering the fact that the cell body itself is probably close to about 50 microns in diameter. So that gives you a sense of how much biosynthetic capacity must be built in to these neurons, as well as some insight as to their need for the energy and the raw materials that's necessary to grow out and maintain the function of that axon. Now, on the function of that axon, this axon has the ability to generate an electrical signal. It's a special signal that we call an action potential, and we'll talk more about that in a later tutorial. That action potential is regenerated along the length of the axon, and in that way it propagates from cell body towards the terminal ending of that axon. So for that reason, we think of the tissue that contains the axons as a conducting zone. It allows for the flow of information from cell body towards axon terminal. Now, at the end of these axons, that's where we find our synaptic terminals, or synapses. And these are specialized contacts that allow one neuron to transfer a signal to another. They come basically in two varieties. We'll say more about these in a later tutorial. There are electrical synapses that have specialized junctions that allow charged molecules to pass directly from one neuron to another, and with it is conveyed the electrical signal. Other kinds of synapses are chemical, which is that there is a space or a gap or a cleft from one neuron to the next, and that cleft is breached by a chemical that's synthesized and released by one neuron, diffuses across that space, and then interacts with receptors on another. So there's a chemical message that mediates the electrical signal from one neuron to another. We'll have a lot to say about synapses, how they work, what are the important regulatory mechanisms that control synaptic activity as we progress through Unit 1. So much more on synapses as we go. Now what I'd like for us to do is to consider how this brief survey of the microanatomy of a single neuron can be compounded many tens of thousands of times in the structure of neural tissue. Now imagine tens of thousands of neurons arranged in parallel, very much like the one we're looking at here. And what I want you to appreciate about this parallel organization is that where we find the cell bodies and the dendrites, that is the input zone of one neuron, we'll find the input zone of many thousands of neurons that are arrayed in parallel in tissue, such as the cerebral cortex. And we have a name for this. We call this gray matter. So as we begin to look into the inside of the human brain, I'll be highlighting for you various divisions of gray matter. Those are distinguished from white matter. And now we can think about the cellular components that help us define what exactly do we mean by gray matter and white matter. So gray matter is where we find the cell bodies, the dendrites, and the synaptic connections of neurons. White matter is the conducting zone. That's where we find axons and the special insulation forming cells that surround those axons called glial cells, specifically in the brain the oligodendrocyte. So that is another component of white matter. And then of course both gray matter and white matter require nutrients in the form of vascular supply, so there are vascular endothelial cells in both kinds of tissue, both gray matter and white matter. And then finally, let me again emphasize that where we find the output zone of one neuron, we find the input zone of many others. So gray matter is both input zone and output zone, depending upon which neuron we have in mind. Okay, well, enough illustrations. Let's look at some actual brain cells. So here are some pyramidal neurons in the cerebral cortex that I prepared myself. And what we're looking at is a stain that shows us beautifully a pyramidal-shaped neuron here. There's its cell body, sort of the pyramid looking something like that, and a thick, stout apical dendrite progressing off towards the surface of the cortex, which is out here somewhere. Here's another pyramidal neuron and its thick apical dendrite. We also see some basal dendrites extending out away from the cell bodies here. Now, we also have an axon, at least one that I can recognize, and it is this process right here. It's extending away some distance away from that cell body. So that's the means by which this particular cell is going to generate electrical signals and then propagate those signals to its neighbors, some quite near, and then some at a great distance away. Here's another pyramidal cell in the visual cortex, but we don't see its apical dendrite because we're looking down from the top, so its apical dendrite is actually right about there. And then here's its cell body, and from its cell body are all these beautiful branching basal dendrites. Now, if we look very closely at these dendrites, and I know this image is not quite as clear as we would want it to be, we can see some spines on these dendrites in that region and here again down there. So if we have a careful look at one such area, I think you'll be able to appreciate that there are in fact some small little mushroom shapes, some filaments that are extending away from those dendrites. And so this would be an example of a typical pyramidal neuron that grows numerous spines along its basal dendrites as well as on its apical dendrite, but typically a little bit further away from the cell body. Okay, here's another view at a large collection of pyramidal neurons, and we can see again their cell bodies. There are little pyramidal shapes here, apical dendrites heading towards the surface of the brain, and then lots and lots and lots of multipolar basal dendrites going off in all different directions. Now we also can detect the presence of axons that are coming out and extending down into this white matter. That's what all these thin filaments are that we can recognize heading down through this relatively unstained background tissue. So here we see all the various components of the functional anatomy of a neuron, the dendrites, the cell bodies, the axons. Well, we can't quite see everything because we haven't yet seen the synaptic terminals. And to give you a sense of that, I want to show you yet a different view of brain tissue. So this is tissue that's stained with a green dye that has labeled axons as they make their way through brain tissue, specifically the gray matter of the cerebral cortex. And I would just draw your attention to these little points of light, these little spots that we can see along many of these axons. So these little points of light are actually places of synaptic contact. Now, some axons, they would grow out a terminal ending, and then there'd be a synaptic junction at that terminal ending. Others tend to make this beaded type of structure, and that's what we see here along these green axons. These are called synaptic boutons, and they're making what we say en passant, or in passage, synaptic connections. So along these boutons there may be synaptic contacts with dendrites out along the sides here. I know I drew them as if they look like they perhaps are spines, but they're really boutons because this is an axon, not a dendrite. So that's what we see here in these little points of light, synaptic boutons. Here's one last view of neurons and their axons. So we see cell bodies of retinal ganglion cells and axons that are growing out away from them towards the optic nerve head. And these are axons that are going to form the optic nerves that allow the retina to connect up with the brain and send visual signals into visual processing centers in the brain. Now let's return to some illustrations of different kinds of neurons and recognize some of the various classes of cells that are present in the nervous system. We've talked a fair amount about the corticopyramidal cell already, and this is a great example of what we call a projection neuron. And the reason why we call it that is because, as I've alluded to, it grows out an axon that can then project for quite some distance within the central nervous system, maybe hundreds of microns, maybe even millimeters, tens of millimeters, maybe even tens of centimeters, depending upon whether the cell is projecting within the brain or whether it's growing out an axon down through the brainstem and into the spinal cord. So projection neurons are called that because they grow axons that project outward away from the cell body over a considerable distance. And these cells often are excitatory, meaning that they have an excitatory effect on their synaptic partners. They make their synaptic partners more likely to generate their own electrical signals. Now, in addition to the long axon that projection neurons send often at great distance away, I'll just point out, as is illustrated here, that the axon can actually give rise to more local branches that can activate cells that are sitting right next to the cell of origin of that axon. So there are local connections as well as a single axon that projects over a great distance. Now let's look at these corticopyramidal cells in context. Here to the left we have our illustration of the individual pyramidal cell, and now we can see the pyramidal cell that is present within the cortex itself, cell body, apical dendrite, basal dendrite, and then a long axon. And here are two other cortical pyramidal cells. So this is a typical view of what a projection neuron looks like and where it might be found in the brain. So the pyramidal cell is a projection neuron. It sends axons over considerable distances that have the effect of exciting their synaptic partners. They may also give rise to local collaterals that might just excite their near neighbors as well. So they sort of have a dual function of activating local cells but also cells at a distance. Now, in addition to these projection cells, we can also recognize some neurons that send axons over a much shorter distance, only perhaps 100 microns or a few hundred microns. One example is found here in this cortical stellate cell. So the cell has a cell body. It's typically multipolar. There are dendrites that go off in different directions. And then there's an axon. And that axon may branch over the nearby territory within the dendritic tree of that cell or just beyond it. So this is called an interneuron because it doesn't project very far at all. So within the same bit of gray matter, we can have projection neurons and we can have interneurons. Now the interneurons come basically in two physiological classes. There are those that excite their neighbors, and then there are those that inhibit their neighbors. And they're roughly equally divided with about as many excitatory interneurons as there are inhibitory interneurons. If we look into the spinal cord, we can also see examples of projection neurons as well as interneurons. Here's an illustration of a very simple circuit that we have throughout our spinal cord and in the sensory and motor elements of our brainstem as well. And this is an illustration of the knee-jerk reflex, also known as the myotactic reflex. So we have two kinds of projection neurons that are involved here. We have a sensory neuron with its cell body in the dorsal root ganglia associated with our spinal nerves, very close to the spinal cord itself. And it has a long axon that runs out through a spinal nerve, and in this case, innervating a sensory structure embedded within the skeletal muscle itself. This dorsal root ganglion cell grows a central process of its axon that enters the spinal cord. Now it's worth pointing out this particular type of neuron to make the following point. This sensory axon is sending signals into the spinal cord. For that reason, we call it an afferent neuron, sending information centrally. There's another type of projection neuron here in the spinal cord, and it's called the motor neuron. It has a multipolar cell body in the spinal cord, and it grows an axon out to the muscle. And the motor axon is sending signals away from the central nervous system to the muscle. And for that reason, we call it an efferent axon. So get comfortable with this difference between afferent and efferent. Afferent means signals coming in to the central nervous system, and efferent meaning signals going out of the central nervous system. Sometimes we use these terms to talk about the flow of information with respect to the cell body itself. We can think of the dendrites as being afferent because information is coming in towards the cell body, and we can think of the axon as being efferent because signals are going away. Now we also have within the spinal cord a short axon interneuron, and that's found right here. So this interneuron has the effect of inhibitoring the antagonistic motor neuron, so that when our hammer tap hits our patella tendon, the quadriceps can contract while the antagonistic muscle, the hamstring muscle, is going to be more relaxed because of the suppressive or inhibitory effect of this interneuron on the motor neuron that supplies that hamstring muscle. Okay, well this concludes our survey of the microanatomy of neurons as well as a brief discussion of different classes of neurons. I hope it's helped you understand the different parts of neurons, what they look like, how they function. So to help you consolidate your understanding of all of this, I'll leave you with just a couple of study questions. Concerning what we've been talking about as the functional microanatomy of a neuron, I want you to think about how does information flow through a neuron? And think about the various parts that are listed out in these response options, and go ahead and select your choice. And then lastly, let's take this concept and compound it and apply it to understanding the structure of neural tissue, specifically in this case white matter. I want you to think about what are the cellular components that we find in gray matter and white matter, and in this question consider those components found in white matter and choose your appropriate response option. Okay, well that concludes our first tutorial. I'm sorry I went on about as long as I did. I will try to be more concise in the future, but I hope you enjoy thinking with me about these topics. I know I enjoy sharing them with you. So on to our next tutorial, considering the non-neural cells that make up the central nervous system. See you next time.